Yeah, thank you very much for the introduction and for having me here today. So yeah, I'll be talking about the evolution of dinosaur eggs and dinosaur reproductive strategies. And so let's get started. Now, modern birds, the only branch of dinosaurs that survived into the present, are generally known for their rather sophisticated reproductive behaviors. They build very complex nesting structures on the ground, up in trees. They brood their eggs. They are very dedicated parents and raise their young. They have different modes of mating systems and parental care. All these different types of very seemingly derived behaviors have been proposed to potentially represent a key advantage that might explain why the avian lineage is the only lineage of dinosaurs that survived into the present. Now, the general problem with testing this hypothesis is that behaviors don't necessarily directly preserve in the fossil record. Now, when we look at the geological record, we're usually left with mostly morphological and anatomical evidence, and reconstructing behaviors from that can be very challenging. Now, Caitlin introduced earlier during her talk the very fact that we're not left exclusively with morphological evidence, but we also can access nowadays, thanks to advanced technology, different kinds of molecular information. So I'm a molecular paleobiologist, and back in 2015, when I started my research career, I thought this is one of the key opportunities, understanding the evolution of reproductive behaviors by using different kinds of molecular markers that indicate them. Now, when we look at the telltale marker for different types of parenting, mating behaviors, nesting modes in modern birds, we have to talk about porphyrin pigments. They basically generate egg color in modern birds, and as you can see here from the background photograph, we have an incredible diversity in terms of egg colors and patterns. And so all this remarkable diversity sort of like captured in this photograph here is based on the different type of mixing of only two pigments. And these two pigments that generate this vast diversity of egg color are called protoporphyrin, a red color pigment, like here basically on the left of my slide, and biliverdin, a blue pigment on the right of my slide. Protoporphyrin and biliverdin, they both represent so-called porphyrin pigments. They're closely related to heme, the red compound in our blood. And they represent, they're directly related chemically. They represent the enzymatic oxidation product of one another. Now, previously, it has not been thought that different types of biomolecule can preserve in deep time. However, if we look at how these pigments are incorporated, there might be a good chance, because in the end, pigments are stored within eggshells, and eggshells preserve very abundantly. Now, here we look at the vertical cross-section of a modern bird egg. And so you basically see at the very bottom the shell membrane, and then on top here, the mineralized part of the eggshell. Now, if we trace how these two different pigments, here in red protoporphyrin and in blue biliverdin, are incorporated in this eggshell structure, we realize that they have quite distinct modes of incorporation. Protoporphyrin is mostly restricted to the uppermost part of what is called the waxy cuticle on top of the eggshell, while biliverdin is highly concentrated in the cuticle, but also extends into the deeper layers of the eggshell. That has generally been attributed to the different solubility properties. Biliverdin is a lot more water soluble than protoporphyrin. However, we've learned recently that that the solubility is not the primary driver for this mode of incorporation. Apparently, we see that once we dissolve the mineral phase in different types of bird eggs, and we're left only with the organic phase, pigments are directly bound to proteins that sit within the eggshell structure and coordinate the arrangement of the eggshell mineral. And so here you see different types of bird eggshells and alligator eggshell on the very top, decalcified, and you can basically see here how the pigments are directly bound to the proteinaceous skeleton of the eggshell. Now, knowing that in the end, eggshell pigments are stored not in the mineral phase of the eggshell, but in the organic phase of the eggshell, that should potentially represent a challenge, again pointing out that it has long been thought that these different types of biomolecules, proteins, and secondary metabolites such as pigments don't necessarily preserve in deepest time. Based on that, I basically started my research with a fundamentally explorative question. Do these different types of biomolecules preserve? Do they fossilize? And can we use them to infer behaviors? And to basically start that quest, I had to figure out what kinds of samples would be most suitable to probe when it comes to fossils. And so here we're looking at an Archosauromorph tree of life. We have modern birds at the very bottom, different kinds of dinosaurs wedged in between where crocodiles go in the tree. And so it has long been thought that, based on observation, that modern birds have colored eggs, while when we look at dinosaur egg fossils, they usually appear somewhat colorless. So it's been inferred that there were no color pigments present. Egg color must be an innovation, and all these very sophisticated behaviors must be an innovation of modern birds. However, is that really true? Now, to basically start exploring that question, I calculated the porosity of these different kinds of eggshells based on a very simple idea. I basically assumed there should be a selective pressure for the evolution of egg color from the moment in time onwards that eggs are exposed to the environment, to the open, to predators, to conspecifics. And with that hypothesis, I basically started sampling. So based on calculating the overall porosity that tells us whether eggs are buried underground or stored in an open nest, I determined that it's the clade of Eomenoraptor or Penaraptor, depending on who you ask, that is basically that evolved open nesting habits and maintained open nesting habits. And so this clade basically includes oviraptors and different other kinds of Maniraptoran dinosaurs, such as troodontids, stem birds, toothed stem birds, and modern birds. And so in 2015, I started this research focusing specifically on oviraptor eggs as the most basal representative of this group of Maniraptoran dinosaurs. And so back then we sampled different kinds of oviraptor eggs from China with a method called mass spectrometry. Here we're doing so-called organic phase extraction of these different kinds of eggshells, and then basically inject this extract and perform reverse phase high performance liquid chromatography, electron spray ionization, quadrupole time of flight mass spectrometry. Sounds very complicated, but it produces these beautiful mass spectra that you can see here at the bottom of my slide. Mass spectrometry works like a very sensitive molecular scale that basically allows us to determine the presence or absence of a certain compound. And here I focus specifically on biliverdin and protoporphyrin. And as you can see here from these two very prominent peaks, we found evidence for biliverdin and protoporphyrin in all of the oviraptor eggshells we looked at. That was incredibly exciting and basically told us, okay, we're up to something here. Now, based on that information, though, we could not really address the question I was really interested in, which is, how did egg color evolve? How did associated reproductive behaviors evolve along the avian lineage? And to answer that question, we obviously needed to sample a lot more types of dinosaur eggs than just the oviraptor eggs that we focused on. However, mass spectrometry is destructive. You powder down your eggshells and you perform an organic phase extraction. And so we basically had to find a non-destructive way to address these questions of whether eggshell evolved once and had a single evolutionary origin, or evolved multiple times convergently. And to do so, I focused on a method called In-Situ Raman microspectroscopy. Now, Raman microspectroscopy is one of these vibrational spectroscopy laboratory tools. Here I'm showing you a couple of photographs from the lab, which basically characterizes the total composition of an unknown sample, completely non-destructive. So with Raman spectroscopy, we basically shoot a laser on a sample and then excite the different types of compounds within the sample that start to wiggle in direct response to their chemical environment. And so these wiggles can be detected in form of peaks and bands in our spectra, and then produce something like a characteristic fingerprint that can either contain or not contain evidence for the two actual pigments we're interested in. Now, the whole problem is, and now we're going back to the one slide that I showed you earlier, that pigments are directly and covalently linked to proteins. Proteins fossilize as well, as Caitlin told us earlier, through oxidative cross-linking. And so at this point, we're faced with a challenge that we're really dealing with very complex heterogeneous bioinorganic materials in fossil eggshells. And so this is not going to be a very simple task to basically tease out the signal related to pigments compared to the signal of other organic compounds. Now, during diagenesis, we know that proteins fossilize through oxidative cross-linking. And while they do this, they basically form these nitrogen heterocycles. And if you look at the chemical structure, this material looks remarkably similar to the different types of porphyrin pigments that we're interested in. What makes it a little more complicated? Now, we found a good solution by basically fingerprinting protein fossilization products from bone that are definitely pigment-free. There are no pigments stored in bone extracellular matrix. And we fingerprinted our two pigments from commercial standards, protoporphyrin in red and biliverdin in blue. And so what you see on top is basically a perfect representation of the summary curve of these three different types of compounds found in fossil oviraptor eggshell that we used for liquid chromatography mass spectrometry before. So we knew there were pigments. And we identified our pigment marker bands in here. With these pigment marker bands, we basically had a set of bands that we would now map out to get a more complete understanding of whether egg color pigments were present in dinosaur eggshells or not. And so with that, we started to analyze all kinds of different dinosaur eggshells. And so there are two different ways of doing this. We've mapped out spots and speckles, and we produced these vertical sections of eggshells, and we traced the very characteristic mode of incorporation of pigment throughout the thickness. As you remember, biliverdin is incorporated in a different way compared to protoporphyrin that sits only at the very top of the eggshell. What we found here is that all of our fossil eggshells produced evidence for protoporphyrin speckling in one way or the other. And then once we started to look at the vertical sections of our eggshells, we realized that information matched up quite well and complemented information on the protoporphyrin speckling on top of the eggshell with background coloration based on biliverdin. We also realized that modern eggshells, modern avian eggshells, and fossil dinosaur eggshells show the same pattern of pigment incorporation into the eggshell, suggesting that to some extent, the mode of pigment incorporation might actually be homologous. The best way to test this is eventually though through phylogenetic analysis, and we'll get there in a second. Now, if we look at fossil eggshells though, you probably realize that there is no actual color preserved. And now that we find evidence for color pigments, we obviously have to ask ourselves, where is the actual color impression? And so what we realized is that through experimental maturation, we can mimic the color change that we observe during fossilization based on oxidative processes that we know occur during fossilization, which seem to not only affect proteins and other kinds of structural biomolecules, but also these secondary metabolites. And so we can basically experimentally recreate the discoloration observed in fossils, which obviously helps understanding why there are no colorful fossils preserved. Now, if we complement in a phylogenetic framework the information that we've just collected, we realize through an ancestor state reconstruction that egg color most probably had a single evolutionary origin in Eomaniraptoran dinosaurs, coinciding with the evolution of open nesting habits, at least based on the current sample set. However, we published these results and instantly got a very good question from a Belgian team that basically pointed out the method that you're using, it's obviously a new approach to the whole topic. Does Raman spectroscopy really tell you whether there was egg color, visibly perceivable egg color, or are you just detecting eggshell pigmentation that would not necessarily cause a visual egg color impression. And so the whole idea is here highlighted with these two illustrations. You can have a white chicken egg that still contains traces of pigments, but is not colored, or you exceed a certain threshold of pigment within the eggshell and then basically cause a perceivable color impression. Now, Raman spectroscopy is non-quantitative, what basically means that is a good question. And so we went back to our data and we sampled all kinds of eggshells, some of them brightly pigmented, some of them barely pigmented. And we statistically analyzed what our Raman signal really detects. And we realized that the minimum threshold of signal required to produce a positive signal based on the analysis we use exceeds the minimum concentration required to generate a visually perceivable color impression. Yay! Now, we worship very critical interrogation of our data. And so we thought, how can we make sure, like that the data that we've collected are really representative of the evolutionary scenario? So we thought, okay, let's look at modern birds and basically go back, evolutionarily speaking, because what we've shown with our molecular analysis of fossils is that birds likely inherited egg color from their non-avian dinosaur ancestors. Now, if we analyze, sort of like going back from the crown to the stem, if egg color was present in the ancestral bird, then we would have independent evidence supporting this idea. And so this is exactly what we did. And what we basically found is by surveying a diversity of egg colors in different modern birds, is that the ancestral bird laid actually colorful eggs, which is of course quite exciting. And so that the ancestral avian egg was very much comparable to a quail egg that you can buy in the supermarket. It would nest on the ground and would show evidence for mostly for biparental care. Now, this is very exciting because now for the first time we have the opportunity to actually infer reproductive behaviors based on egg color as a behavioral proxy. We can make inferences about the mode of parental care. We can make inferences about the reproductive season, the humidity during the reproductive season. We can even make inferences about communal, so shared nests, and the potential presence and competition with brood parasites. But if we look at these data, there is one question left. Why do we find egg color only in these Eomanoraptoran dinosaurs? Why were sauropod and ornithischian eggshells not colored? Is there anything peculiar about theropod eggshells? Indeed, there is. We recently discovered two different types of eggshells, ornithischian Protoceratops eggshell, as well as early sauropodomorph Mosaurus eggshell, that were in fact soft-shelled. And so here you see the clutches. The Mosaurus eggshell material and the Protoceratops material both have embryonic evidence, so we can assign them with confidence. Now, if we analyze these eggshells chemically, we basically realize that their organic eggshell lining is endogenous in composition, so it's very different from organics in the sediment. And then when we read these data into a training data set that allows us to distinguish on a molecular level hard-shelled eggs from soft-shelled eggs, we realize that here in red outline all our fossil data points for Protoceratops and Mosaurus plot among the soft-shelled eggs. That's very exciting. And so once we now combine all this information, we basically realize that here I'm adding information for Protoceratops and sauropodomorphs. The hard-shelled egg evolved 3 times, at least 3 times, independently. One of these independent lineages of calcified eggshell is the theropod lineage, the lineage in which we find evidence for eggshell coloration and all kinds of sophisticated reproductive behaviors that once were thought to be unique to modern birds. Now, with this information, we basically realized that indeed, the avian evolutionary lineage, the avian eggshell, avian reproductive behaviors are unique among other types of dinosaurs. And might in fact be a reason, one of the main reasons, why birds are the only lineage of modern dinosaurs that survived into the present. Now, no science story is ever truly concluded, and so I thought I'm going to close out this talk with a little teaser where we're going next in terms of research. One of the big questions that is now coming up and left here is, of course, birds brood their eggs. And in dinosaurs, that is a behavior very difficult to trace. One would have to have actual information about their thermophysiology, about their thermophysiological strategies. Very difficult to trace. Now, if we wanted to understand which dinosaurs were capable to brood their eggs and which ones were not, we would need that kind of information. Is brooding an avian feature, or did brooding, as well as other supposedly avian features, evolve in the dinosaur stem? This is the big question we're targeting at the moment. And I'll just leave you with this. Stay tuned. I have to thank a bunch of collaborators who have been working with me on these different kinds of projects that I've been sharing with you today. So thanks to all of them. Thanks to all funders. Thanks for your attention. I'm happy to take any questions. Also, feel free to reach out via email.
Just a final reminder, if anyone's watching online, they can put their questions in the chat there and we'll take questions from the room as well.
So what purpose does egg coloration serve for the parents, for the animals themselves? Like why do they have colored eggs?
That's a really good question. So it really varies from species to species. Generally, egg color serves primarily visual purposes. In many species of birds, the egg color sort of matches the tone of the nesting background, where then the key function is camouflage, right? So whenever there are visual predators such as other birds or mammals, it is definitely beneficial for birds to color the eggs, which is an energetically costly process, physiologically speaking. It is beneficial for them to color the eggs to somewhat match the tone of the nesting environment so that the eggs don't necessarily stick out to any visual predators. But then there are also other kinds of more derived and sophisticated behavioral adaptations related to egg color. So it has been demonstrated that especially in certain types of birds, such as, for example, blue-footed boobies, there is sexual signaling going on, where eventually the color, and it's specifically related to the concentration of biliverdin, so the blue pigment in eggshell, where the concentration of blue color pigment within the eggshell signals to some extent the quality, the health, the survival potential of the offspring to the male parent. And so these are usually monogamous mating systems, where then basically both female and male brood and raise the chicks. And so eventually the bright blue egg color is generally associated with some kind of motivation to the male to provide the best possible paternal care. We find blue egg color in all kinds of dinosaurs, and it's quite exciting to see that our egg color analyses match up very nicely with independent analyses of the evolution of paternal care. So we find sort of like support for ancestral paternal care in dinosaurs based on statistical evidence, as well as based on egg color.
So I know in certain modern avian species, you will see the presence or absence of a brood patch to indicate brooding behavior. Have we seen, I know that fossilized skin and feathers are relatively rare. Have there been enough fossils examined to maybe look for something like this as an indicator of whether or not dinosaurs exhibited brooding behavior?
Yeah, that's a really good question. I don't think anyone has actually surveyed obviously we would need exceptionally preserved fossils like the Sinosauropteryx that Caitlin showed earlier. We would, yeah, I mean, I guess no one has in detail surveyed the sort of like patchiness of feather preservation, which is certainly something people should look into. I guess it would be very difficult to distinguish taphonomic biases that act potentially even within one carcass from sort of like the seasonal like local loss of feathers that then eventually create the brood patch. But yeah, that's definitely an exciting question that someone should look into at some point with these exceptionally preserved fossils that we have from some kinds of Lagerstätten.
Do you think this indirectly proves that at least many raptorians had color vision?
Yeah, so, well, absolutely. We know that egg color serves primarily visual functions. And if we look at the evolution of color vision, of color pigments, there is a very exciting perspective out there in science by Koschewitz and colleagues that are basically reconstructed independently that tetrachromatic color vision may actually be ancestral to most kinds of panoraptoran dinosaurs. So yeah, I would definitely treat it as evidence for the ability to see color, whether trichromatic or tetrachromatic, likely tetrachromatic because that's the mode of color vision present in most birds.
All right, so we have another question in the chat that's from Dr. Mark Lowen. Just amazing stuff. How much are you expanding your search deeper into the theropod and ornithischian tree for both color and shell types?
Oh, that's a very good question. Well, so for the shell types, I think that's the obvious one. For shell types, we're very actively expanding the data set at the moment, and there will be soon very exciting news about different kinds of sauropodomorphs. We've looked at Massospondylus, Mosaurus, Laphangosaurus. We have a very exciting clutch of potentially soft-shelled Lambeosaur eggs, embryo-bearing as well. So there is definitely like a lot of exciting eggshell material coming out there. In terms of egg color, it's obviously more complicated because we need carbonaceous preservation for most of these analyses. And when we look at what is happening at the sort of like in the Maniraptoran stem, the coverage with unarguably assignable eggshells that meet this criterion is not too good. But there are definitely eggshells out there that I'd like to target in the future, and we're currently working on Tyrannosaur material.
Does the texture, can you hear me?
Yes.
Okay, sorry. Does the texture of the eggshell have anything to do with whether the parent turns it during its brooding?
Oh yes. So very, very good question. So if we look at modern bird eggs, for example, emu or even ostrich eggshells, brooding leaves very clear traces when it comes to the abrasion of ornamentation. And so in ostrich eggshell, we don't have much ornamentation, but after brooding, eggshell looks definitely very, very polished. And so in emu eggshell, it is even more obvious because there we have very strong ornamentation in freshly laid eggs that is then basically like eroded during the brooding process. And so the different types of sort of like brooding of rotating behavior, they leave characteristic striation orientations on the eggshell surfaces. I don't think anyone has surveyed this in detail in fossils. Taphonomy is of course a factor to consider here because many eggshells are not necessarily preserved like in situ without any transport. So quite often we look at eggshell assemblages where the material has been transported quite a bit, and then you induce these kind of microscopic scratch marks and striations just based on the sediment transport and the contact with siliceous grains in the sediment. But for in situ preserved eggshell material, that is definitely something people should look into.